For most of us, if we could just act on what we already know about health, finally change those 2 or 3 basic health habits we've been meaning to change, we'd experience a dramatic improvement in overall well-being. But knowing what to do and actually doing it are two very different things. We need an approach that supports true personal health transformation. And this is why I launched a new membership-based health service called Adapt 180 Health. As a member, you still get answers from licensed functional medicine practitioners and even custom nutrition plans from licensed nutritionists, but it doesn't stop there. You also get your own board-certified health coach, an expert in lasting behavior change. Get the support to truly transform your life and unlock more health, joy, and resilience. Visit adapt180health.com. That's adapt180health.com to learn more and download our free ebook. Hey everyone, Chris Kresser here. Welcome to another episode of Revolution Health Radio. This week, we're going to be talking all about COVID-19 testing and what the currently available testing can and cannot tell us. There are a lot of misconceptions out there right now about testing, whether we're talking about PCR tests or rapid LAMP tests or rapid antigen tests. And I wanted to record this episode because I know we're approaching Christmas holidays and a lot of folks are considering traveling and thinking about what role testing might play in determining if traveling to see relatives is safe. And there is just a lot of misinformation out there. I have been confused about it myself, but I invited Dale Harrison as a guest to discuss this, and I hope that this episode will clarify a lot of the misconceptions and help you make a more informed decision. So Dale Harrison is a senior executive with more than 20 years of experience in the biotech industry. He's held multiple VP and C-level positions with companies in the research tools and molecular diagnostics sectors, with direct experience in PCR, next-generation sequencing, CRISPR diagnostic platforms, and assay development. His original background was in research physics and later technology development around robotics and automation. He transitioned into the biotech market in the mid-1990s, originally working for a VC-backed startup as CTO, developing robotic DNA and protein synthesizers and high-throughput automated production lines for DNA synthesis. Since then, he's gone on to hold CEO, CTO, and VP of commercial development roles in multiple companies in the biotech space. So Dale is super knowledgeable, not only about the underlying scientific methodologies that are used by these different testing platforms, but also on how the performance of these tests differs in the real world versus, the studies that are done on them in highly controlled environments. And that's going to be a big theme in the show, as you'll see, because it really changes things. That if the test doesn't perform in the real world as well as it did in the lab or in the validation studies, that's information that people really need to know about, whether you're an individual or you're a clinician that's running these tests. So I hope you get a lot out of the episode. It's a little bit technical in parts, so bear with us, but there's some really solid info in here. And again, my hope is that it helps you to make a more informed decision about your gatherings over this winter. All right, so without further delay, I bring you Dale Harrison. Dale, it's such a pleasure to have you on the show, and thanks for agreeing to join us on such late notice.
Well, thanks for inviting me.
So let's start with your background in molecular diagnostics. We're going to be talking all about diagnostics and testing for COVID today, and I'd love to give people a sense of where you're coming from.
So I've been in the biotech industry for about 20, 25 years now, since the 90s, and mostly around various aspects of molecular diagnostics. So I've worked in what's called oligo manufacturing, where you manufacture the synthetic DNA that goes into some of these molecular tests. I've run a genetic sequencing lab. In more recent years, I've focused much more in the area of clinical diagnostics, and with work in areas like next generation sequencing as a diagnostic platform, as well as PCR and LAMP, and currently involved in a project using CRISPR technology to do viral diagnostics, and some experience on the FDA regulatory side. So
Great. Those are all very relevant to the conversation today. We're going to be talking about PCR LAMP, a new CRISPR test that was just, I mean, it's not available yet, but there was a study just published on it, and then some of the finer points of these different types of tests. So I want to start with like a 30,000-foot view, which is the difference between test performance in a laboratory or perhaps in a very well-controlled scientific study and test performance in the real world when real people are out there getting these tests. This is something that you've mentioned in a couple of your articles on your Substack blog, which we'll talk about at the end of the show, and in email correspondence back and forth. And I think it's really important for people to understand. So tell us a little bit about that.
So when a test is approved by the FDA, there are a set of documents. There's a key document called an IFR, an instruction for use document, IFU document, and in that it usually contains some information about the performance of the test in terms of false negatives and false positive rates. Then you move into the real world. The real world tends to be a great deal messier. But one of the things to understand about these sorts of tests in general is that certainly for infectious disease testing, what you're looking for is some molecular fingerprint of a particular pathogen, a particular virus or bacteria, or there's a number of other types of pathogens that you can test for. And so generally what you're looking for is either the presence of a highly targeted protein molecule or a highly targeted DNA molecule that would only be present if the virus or the bacteria was actively replicating inside a patient.
Mm-hmm.
So one of the problems with testing in general is that it's not entirely a clear yes/no answer. So you can get you can have a test and get a positive result, and that positive result can be an error. It can, so it can come out with a positive even when there's no infection. It can also come back with a negative when there is an infection. Now, with most testing that's out there at the moment, the issue is much more around the false negatives. The false positive rate is extraordinarily low. And again, if you look at these FDA documents, what you see is, typically 99+% specificity, meaning that it can reliably give you a positive answer if the pathogen is present. So the tests are really quite sensitive and quite specific. The problem comes in when you actually try to deploy this in the real world, because the validation studies that go into the FDA approval are typically done in pretty ideal conditions. You're selecting the particular patients that you're going to test. You're selecting the conditions that the patient is in. And you've also got typically pretty highly skilled people performing the test, gathering the samples, running the instruments.
Right.
Once you move into the real world,
in a controlled environment, right? They're not standing in a parking lot,
right,
reaching into someone's car and swabbing their nose, right?
So when you move into the real world, a lot of things can go wrong. And what you end up seeing is that almost everything that goes wrong will ultimately result in what's called a false negative, meaning that the test returns a negative when in fact you're infected. And the false negatives are really the serious issue with these tests. So, one of the things is that the tests tend to be very sensitive to how well the technician taking the sample does their job. This is something that requires a real skill and precision to do it. So these technicians are generally very well trained, but if you're doing, taking a lot of samples in a short period of time, again, there's a lot of things that can go wrong where the person taking the sample just doesn't quite do it right. And so you end up collecting enough sample to be able to be detected by the test. The other thing that happens is on something like COVID, it's an upper respiratory disease, the virus will typically colonize the nose and upper throat first. And then as the infection progresses, it'll tend to migrate down lower into the lungs. So if you're pulling a sample just from a nasal swab or a sample from a throat swab, if the virus has already migrated lower, you may simply miss it. The other thing that happens is there's a process called mosaicing, where not the virus isn't necessarily spread evenly across the entire nasal cavity or the entire back of the throat, that it appears as though it exists in patches where this interspersed by patches where there's very little virus. And so if you just randomly hit the wrong spot, you end up getting, in some cases, too little virus in the sample to be able to detect.
This has been an issue with H. pylori testing as well, and other pathogens that have that same patchy distribution.
Yeah. And the other issue is that once the sample's collected, it's got to be properly handled, properly packaged, properly shipped, and properly labeled. And so there are a number of things that can go wrong. So one of the things that happens is that most of the things, especially for viruses, most of the viruses that we're looking for, when you take the sample, with that sample comes other enzymes that are present in the nose or in the throat, whose job it is to break down RNA. This is especially an issue with RNA viruses. So flu, COVID are major examples. So these enzymes will very rapidly break the RNA down. And so the sample has to be transferred into what's called a viral transport media, which is a special reagent that basically slows down that enzymatic process, slows down the degradation of the RNA. The tube needs to be properly labeled, and it needs to be shipped. And so, hopefully all that goes well. And then once you get it at a testing lab, typically you're processing hundreds or thousands of samples at a time in large batches. And most of this is done robotically. And so, there are additional areas of where errors can occur just in the robotic handling. Now, all the things I've talked about so far are really things that will cause you to miss getting enough sample to detect. So you're going to get a false negative. The one area where you actually see a very, very low rate of false positives, and we're talking, one sample in 10 to 100,000, is basically error handling by the robots, where you're mixing reagents on a plate. So typically you do these things on what I call 96 well plates with about 92 samples and about 4 controls on the plate. And the robot has to add reagent, has to mix reagent, and you can get cross splashing if the robot gets a little too aggressive with it. And there was actually a case back in May and early June where one of the major tests was seeing false positives at the rate of about 1 out of, again, between 1 in 10,000 and 1 in 100,000 samples. And it was traced back to cross splashing between the wells because the robot had too aggressive a mixing protocol.
Right.
The FDA issued a notice. There was corrections made to the software, and the problem went away. But the risk of getting a false positive is very, very low. It's generally means that you've either mislabeled a tube or you've accidentally had contamination from one well, from one sample to another sample when it's on the robot. So a lot of there's a lot of nonsense talk about you can't trust the test because it's a lot of false positives. And the fact is there are no false positives. I mean, they're effectively zero.
Right. With the exception of maybe antibody testing, which we can talk about a little bit more later. But, if we're talking about PCR and, testing for the actual pathogen, it's the specificity is so high that effectively, as you're saying, the false positive is not really the thing we need to be concerned about. It's much more false negative, which of course carries more risk too. The risk of a false positive is that someone unnecessarily has to self-isolate, and that does have some, significant downsides, but not to the extent perhaps that a false negative does when someone is actually positive and then believes that they're negative and maybe isn't as careful with distancing or wearing a mask or other measures that they might take. And from what you've described, it's pretty clear that there would be a significant difference from carefully controlled test environments and the real world. It's like I said before, you imagine here where I live, it's somebody who's standing in a parking lot, outside of a hospital. It's cold. They're standing there for a long period of time. they are probably very well trained, but just people are human, having to do that many swabs, you've got a lineup of cars and having to do that over and over again while you're standing outside for a long period of time. It seems almost impossible that there wouldn't be some errors introduced in that process.
Right. And again, there's this difference between the official false negative, false positive rates, the official specificity and sensitivity rates, and then what your effective false positives and false negatives are in the real world. So
Right.
again, things like the robotic handling errors I was talking about, that's something that really has nothing to do with the test. It has to do with how you handle the sample.
Sure.
The errors around sample collection or just the randomness, has nothing to do with the core test. So the thing that people need to understand, and even a lot of people in the clinical environment fail to really understand these distinctions, is that the information from the FDA that comes from the test validation process really represents the absolute ideal performance of the test. And then everything that happens from that moment forward is going to be worse than that level of performance. So one of the analogies I make is, you go to the car dealership and the dealer tag on the car says that you should get 45 miles to the gallon. You buy the car, but the reality is you never see more than 28 miles to the gallon.
Right.
And it's to the point where the TV ads all end with "and your mileage may vary." Um, and again, because what's happening there is in the regulatory environment, how miles per gallon is measured for a vehicle is in an extraordinarily idealized environment.
In a wind tunnel or something,
Right.
inside
And
a factory somewhere.
and it represents, the sort of the ultimate upper ideal performance of that engine, but it doesn't really represent what you see in the real world.
This is so important because I have, a number of patients, family members, friends, and practitioners that we train in our training programs, reaching out and they go to, Abbott's website, for example, and they look at the specificity and sensitivity numbers for, well, let's just focus on sensitivity because as we established, the specificity is not really much of a consideration, but they look at the sensitivity numbers for the Abbott ID Now or the Abbott Binax Now, which we'll talk about in a moment, and they say, "Hey, these look pretty good. This seems to be a pretty reliable test." But what you're saying here is pretty clearly is that we really can't look at those numbers and assume we're going to get that kind of performance in the real world. And the difference is not insubstantial. We're not talking about like 1 or 2%. We're talking about, you know, north of 10%, maybe even 20% difference in the numbers.
Or even greater in cases.
Right. So let's now switch gears and talk a little bit about the currently available testing options. If we can kind of break it into three broad categories. One is PCR, is the rapid testing, and then one is antibody testing. So give us an overview of each of these different types of tests.
Probably should break it into four large categories. So PCR, rapid test, antigen test, which is a different type of rapid test, and then the antibody test. And so
Right.
the question is, what are you testing? You have tests that answer the question, do I have a current active infection? Meaning, is there virus that's replicating in the patient? And then there's a second type of test that answers the question, was I previously infected? And so let's start with the was I previously infected? These are the antibody tests. And what they're looking for are, as the name implies, they're looking for antibodies in the bloodstream. So what characterizes them is that you're doing a blood sample, not a nasal swab or a nasopharyngeal sample. You're either doing a full blood draw or you're doing a pinprick. And so you're looking for protein molecules that your body's immune system has manufactured to attack the virus. And these generally won't begin to show up at detectable levels until about 2 weeks after the onset of symptoms. So this is not something that's useful, during the time that you're sick, but it's useful to look back, retrospectively to know whether or not you've been exposed. The other types of tests are really looking at, is there some sort of an active infection? And so the three big categories there are the PCR test, which is, they go in, you go into a clinic, they have the, foot-long Q-tip that they shove down your nose. They're actually gathering a sample from the upper back portion of the throat that can't be reached from the mouth. They're actually just going through the nose to get to the throat.
Yes, they're not pleasant.
Yes. Well, there are even more unpleasant versions that have been pioneered in China. That's an upper respiratory. That's more like
Oh, wow.
it goes down the throat into the upper portion of the lungs, and it provides much more reliable results, much lower false negative rates. But it's sort of like, a cross between sword swallowing and
Right.
using a Q-tip.
Might be hard to get people to come get tested once the word gets out about that
Right.
test. Yeah.
Well, that's why it works in China, because you don't get
right. No choice. Right. So, okay. So we've got PCR. That's one of the three.
And that has to be sent off to a lab. Under ideal conditions, you might get the result back in a day. Under realistic conditions, it's going to be more like 3 days to maybe a week. If things are really bad out there, it can stretch to as much as 10 days or 2 weeks, simply because the labs are backed up. Then there's another category of tests that are just generally called rapid tests. And the sort of the classic one is the Abbott ID Now. This is the one that was famously used by the White House and helped give us the Rose Garden super spreader event.
Right. Was NBA also using that too, I think, in Florida when they had their,
Yeah. And so this is a test that's done on site. So it's a so-called point of care test, meaning that the sample is gathered, the instrument is right there where the sample is gathered. You prep and place the instrument the sample directly on the instrument, and you get an answer back quickly. Quickly meaning 20 to 30, maybe 40 minutes at the most. And then this third category, antigen test. So what the antigen test does is that it's not looking. So these first two tests are looking for genomic RNA from the virus. So part of the virus's genomic code. The antigen tests tend to be even faster. These are 5-minute, 10-minute tests. They usually use what's called a lateral flow strip, which is a little disposable plastic thing that looks like a pregnancy test. Matter of fact, a pregnancy test is a type of antigen lateral flow strip test. So these antigen tests are not looking for RNA. What they're looking for are proteins. And specifically, everybody's seen a picture of the virus with the big ball with the spikes on it.
Spikes, yeah.
So inside that ball is another ball, or more like a bag. So there's a bag made out of proteins called the nucleocapsid. And that bag contains all the RNA. And so the nucleocapsid tends to be very unique to a specific virus, where a lot of the proteins on the outer coat can often be hijacked from other proteins the cell is manufacturing. And so you have to be careful about trying to test for what's on the outer coat, the outer envelope. What you need to look for is the nucleocapsid envelope inside. And so these are just proteins. And so these tests are looking for the presence of these proteins. So the advantage of these is that they run very fast. I mean, again, many of these you get an answer on the spot in 5 minutes. The downside is that they tend to have a very, very high false negative rate. So in testing, molecular testing in general, there's this idea of no free lunch, that you can have a test fast or cheap, or having high specificity and sensitivity, meaning very low false negatives and very low false positives. So basically you get one of the three. And if you give up one or more, you give up one of those items, you can then gain in the other areas. So with the rapid test, the 20-minute test, what you're essentially doing there is it's still a relatively expensive test, but it's a fast test. And so that speed is paid for by having less ability to be able to detect the pathogen that you're looking for. The antigen tests are both fast and cheap. And, so things like the Abbott Binax Now test, which is a, very inexpensive $5 to $7 one-use disposable test. And you can run the whole thing in 10 minutes. So there you've got fast and cheap. but you pay double for that in terms of very, very high false negative rates.
Right. So let's actually bring that into more focus here now, since we've given a kind of overview of each of these tests, and we're going to do a little bit of a deeper dive into each of them in a moment. But what can people generally expect for false negative rates with these tests in the real world, not in in carefully controlled conditions, but in the real world? So PCR, the rapid test, the LAMP, and the antigen test. What kind of false negative rates might we see?
So there've actually been some studies that have made it into the peer-reviewed literature looking specifically at this question. And again, there are a lot of variables. Are you doing it on a drive-through line versus in a major medical center? But in general, the what's called the laboratory run PCR, which is the sort of the standard test that everyone thinks of that gets sent off to a lab and it's a few days to get the results back. Those are going to typically run about 20% false negative in actual clinical usage.
Right.
The rapid test that you do right at point of care is higher than that. It's typically going to be 30 or 40% false negatives. And I'll give you a specific anecdote on that. So I have a relative who is an air ambulance medical technician. And so they have one of these Abbott rapid tests on board the helicopter because, they go to do, a cardiac patient or they go to do a car wreck and they need to, rapidly know should they don all of their
Protective
protective
PPE.
gear.
Yeah.
And what they were finding was that repeatedly they would pick up a patient unrelated to COVID, car wreck, a motorcycle accident, a cardiac event. They'd test negative in the helicopter. They'd get into the ER. They'd be able to run a better test, typically a full PCR test, because a lot of hospitals will have a lab
Have their own equipment there.
for internal use. And so they can turn these things around, in an hour or so.
Right.
And they were repeatedly finding patients that were testing negative in the helicopter are now positive when they're in the ER. And what that led to was them basically abandoning the use of the rapid test and just always flying in full PPE.
Right. Just
Which is
assuming that everyone is positive, basically.
right.
Yeah.
So because that was the only safe way to transport patients was just assume everyone's a positive.
Right. So
Um,
that's pretty high, 30 to 40% false negative, but it's even worse for the antigen, right?
right. And so these antigen tests, the reality on these things is that they're about a 50%, sometimes more than 50% false negative rate. They can be quite high. And again, there's a lot of reasons for that. Some of it having to do with sample collection, some of it having to do with processing the test, especially on the tests that they don't have any that are legally allowed for home use yet, but ones that are getting ready for home use that are designed for a non-trained person to be able to run the test. You've got a lot more opportunities to get things incorrect there. So there are a number of issues with that, but there's a more fundamental issue with these tests. So if you drop back for a second and ask the question, what are these tests actually doing inside the machine? And the way these tests work is that you've got a very specific target molecule. So for the PCR test, it's generally a very short, maybe 50 nucleotide long RNA sequence that is highly unique to that virus that won't be found in any other organism on the planet. And this allows them to be very specific, to be able to make sure that if they detect that thing will only have come from the virus that they're targeting in the test. But the way these things work is through a process called molecular amplification. So it's essentially a chemical process that allows you to make millions, billions of copies of a particular molecular sequence that's found in the sample. And so a laboratory PCR test will do typically about 40 what I call cycles. Each cycle doubles the number of copies of the targeted sequence that was in the original sample, which means at the end of this, you can get between 1 billion to 1 trillion fold amplification, but you still have to have some material to start with. So if you don't have any of the targeted viral RNA in the original sample, you could run this through 500 cycles and you're going to get nothing.
A billion or a trillion times zero is still zero, right?
And that is why these tests have such a low false positive rate, is that they're able to go in there and take very, very tiny amounts of sample and amplify it enough that they can detect it at a macroscopic level. What they're actually doing is they amplify these things. They're attaching fluorescent molecules, and then you basically shine a light on the test chamber that contains the liquid that's going through this amplification process, and those fluorescent molecules will then light up, and you can detect that in a very specific photodetector. So you're looking for a very specific wavelength of light shining off of the molecules. And once you've got a large enough signal for your photodetectors to see, then you've got a positive, and you actually typically will stop the run at that point because you've got a positive. The LAMP process still uses so these are the rapid tests, the 20-minute test.
Mm-hmm.
They still use molecular amplification because again, that's the only way to really get a sensitive test. But part of what makes them rapid is that they're using both a less efficient amplification technology, and they're running it for a shorter period of time. So again, no free lunch. In the end, what happens there is that you've got 1,000, maybe 10,000 fold lower level of amplification at the end of the test than you would have with the regular PCR test. And so you simply have less sample to be able to detect and a greater likelihood of missing what you're looking for, therefore higher false negative rate.
Right.
The antigen tests are unique because they're not looking for DNA or RNA. What they're looking for is a protein. And the problem is, we know how to amplify DNA. We do not know how to amplify proteins. There are ways to amplify proteins, but it's something that looks more like a long-term fermentation process where we're actually growing organisms over
Right.
long periods of time. So there's no rapid chemical molecular amplification technology for proteins the way there is for DNA. As a little side note, so the COVID virus is actually an RNA virus. We also don't know how to amplify RNA, but we do know how to amplify DNA. And so these tests are often referred to as RT-PCR tests. The RT stands for reverse transcription. And so on the very first cycle in the test, you essentially turn the RNA into a matching DNA sequence because you can amplify the DNA, but you can't amplify the RNA. And so you basically it's called reverse transcription, because in a normal cell, the DNA in the cell transcribes to RNA, which then translates into proteins. This is sort of how the blueprint gets executed from DNA into proteins. Right. So reverse transcription is reversing that process in the opposite direction from what normally happens in a cell, going from RNA to DNA. With the antigen test, because we're looking for protein molecules, there's no amplification technology for proteins that can work on a pure chemical basis rapidly within a test. And so you rely on a different sort of amplification process where you're essentially binding antibodies to the antigen and then flowing that complex down a paper strip typically, and having that antibody trigger a whole series of molecules that are attached to the surface of the paper that will cause a color change. So there is a type of amplification, but you're not actually amplifying the original protein molecule, but you are amplifying the effect of it as you flow it down a strip of paper. And again, this is how a home pregnancy test works. Right. So the end result there is you have something that's very fast, that once you put the sample on the little lateral flow strip, you'll have an answer in 5 minutes or less. But you've got a very low level of amplification, and therefore you need a lot of virus in your sample in order to be able to detect anything.
Right. So
And
this is probably a good segue into the another weakness of the antigen test, is that that's not approved or validated for people without symptoms. And that I think the one study that I had sent you or you sent me showed up to a 70% false negative rate in people with no symptoms.
right, right. So right now, the tests that have what's called EUA, Emergency Use Authorization approval from the FDA, have only been validated on patients that are symptomatic and are approved for patients that are symptomatic. Meaning, and again, the idea of the antigen test is, you're a physician in a hospital, you've got the patients in front of you, you've got all of their symptoms, maybe you have lung imaging, you're seeing clear indications of COVID. You need to confirm your diagnosis because you need to either send that person to the COVID ward or to the pneumonia ward. And you don't really want to make a mistake there, but you got to make a decision now. And so, the original idea anyway, behind the antigen test was that you've already essentially got a medical diagnosis. You've got all the indicators there, and you want to confirm that diagnosis with a very quick test because you have to make an immediate decision.
Right. So
Um,
very different than a screening test of people
right.
who are asymptomatic.
And the reason, so what happens is when you start using the test on asymptomatics, it's not universally true, but most people who are asymptomatic are in fact what are called pre-symptomatic, meaning they will eventually develop symptoms, but the virus hasn't replicated to the point that the immune system recognizes the virus. And
So it's
because
not producing those antibodies that will bind to the antigen and show a positive result.
well, no, because again, this is measuring proteins from the virus itself, not
Right.
antibodies.
Right.
But what happens is you just don't have enough virus. You don't have a high enough viral load at that point because again, there are some people that appear to have a very high viral load, are very infectious, but never show symptoms. Then there are other people that are the pre-symptomatics, which is more common, where you've got a high enough viral load that you're infecting people, but things haven't progressed to the point that the immune system is aware that there's an infection and has started to kick in. And so as the virus replicates more and more, it finally reaches a point to where there's so much virus present that the immune system sees it. And so the antigen test is useful when there's a very high viral load, because again, ideally you need a lot of virus collected on that sample in order to have enough proteins, viral proteins, for this fairly insensitive test to be able to see anything. Now, what that means is if that antigen test returns a positive, you can take that to the bank. That's absolutely guaranteed 100% positive. And this is one of the things that's interesting with the antigen test is that there are a lot of sort of handling mistakes that can be made with a PCR or even a rapid test that because the antigen tests are so simple that you've got fewer, it's not going to get left in the FedEx truck in the sun for 2 days. It's it's not going to get mislabeled. So a lot of those sort of handling mistakes that are issues with the other tests aren't really an issue with the antigen test because it's immediate, it's right there in front of you. So if you get a positive on an antigen test, you can absolutely take that to the bank. It's absolutely for sure you're infected. If you get a negative, then you pretty much have to assume that you're a presumptive positive until you can get, a better test to show a negative.
Right. So a positive is a positive, a negative is a maybe.
Yeah, a negative is a presumed positive.
Right. Yeah, even
At least for
stronger.
the antigen test.
Right. So let's also talk about test timing because that's another issue even with PCR and the lab testing is let's say you get exposed on an airplane, you're going to visit your family or something, you get exposed on an airplane and right when you arrive at your destination you get a PCR test. Is that likely to return a positive result even if you've been infected?
Right. No, it won't. And so typically if you assume it's sort of a normal progression for COVID, from exposure to onset of symptoms to essentially clearing the virus from your system. For most people, and again, there's a lot of variability here, but for most people, that's about a 10-day cycle. Half of it you're pre-symptomatic, half of it you're symptomatic. So on average, most people will take about 5 days between when they got infected and when they start to show symptoms. They start to run a fever, for instance, or have a cough. And then it actually only takes about 5 days after you start developing symptoms before the immune system has effectively cleared the virus from your system. And so what happens is that you need the virus to replicate, to grow for a day or 2 days after you've been infected before a test will reliably pick up a positive. So, if you get infected this morning and you get tested this afternoon, it's probably going to be a negative. If you got infected 2 days ago and you got tested today, then you've got a pretty good chance it's going to show up a positive.
What is the correlation between infectiousness and the likelihood of testing positive? In other words, some people have said, well, hey, if you test negative, that means there's not enough virus replicating to show a positive result, but it also means that you're likely not contagious. Do you think that can be accurately said or not?
No, there's absolutely no reliable evidence. And I'll just as an aside, so I work with, I have clients that are a number of companies in molecular diagnostics. I can tell you every single one of them would love to be able to produce an infectiousness test. And no one has any idea how to do that. What these tests do is they answer the question, is there replicating virus in you? But whether or not there simply isn't enough known about how much virus, where the virus needs to be. There's a lot of unknowns around what causes one person to be infectious versus another person. One of the things we know is that almost all people who are infected infect none or maybe one other person. And a very small number, maybe 15%, maybe 20% of the outside are responsible for 80 or 85% of infections. And so
super spreaders.
These are the super spreaders. And no one can tell the difference between a super spreader and a non-spreader. That, in terms of anything you can measure, look at, test, they don't look any different. But that super spreader, might be passing the virus on to 30 or 50 or 100 other people, where, most of the people with COVID, may pass it on to none or to one person, maybe a couple of people.
And that's the real wild card here. We have no way of distinguishing between someone who's very unlikely to spread the virus and someone who is a super spreader. So again, it's kind of similar to a negative is a presumed positive. I guess you kind of have to presume that somebody is a super spreader unless you have evidence that they're not. And we don't have any way of, at this point, showing that they're not, right?
Now, there is some assumption that they must be expelling a larger amount of virus. It doesn't mean they have a larger amount of virus. They just may be more efficient at expelling it. There may be something unique about the super spreaders where, every breath, every time they breathe out, they're somehow moving much more of what would otherwise be the same amount of virus, into those droplets that they're breathing out, into that aerosol that they're breathing out. So the problem is we just don't know what that is. And if you don't know what it is and you can't characterize it, you can't develop a test for it. The other issue is there's so much variability in the sampling. So you could take the same technician and the same patient and do 10 swabs in a row, and you're going to see quite different quantitative results in the test run. So again, this is this nonsense that's been going around for some time about lowering the cycle threshold. So this is the number of amplification cycles that the PCR process is going through to amplify the original RNA from the sample. And the problem is you can't say that 30 cycles means that you're not infectious to others, but 40 does, because you could see you see huge variances in the amount of virus that gets collected in a sample, again, with the same technician using the same technique on the same patient multiple times. And again, I've looked at some of the data from the validation studies, that are done, as a part of submitting the FDA application for approval. And there's just huge variability. And that's why these tests are generally binary tests. They give you a yes, no answer. There's not useful information in the cycle count for the most part.
Right.
Certainly not something, not useful at a diagnostic level.
Not to the point that we can rely on it and make decisions based on that information. So let me just summarize what we've talked about so far. Then I want to kind of make this a little more real by talking about like some, a scenario like someone going to visit their family. And then I want to just finish up by quickly talking about new test methodologies that are either in development or maybe even being close to being released that might be able to help address this quagmire that we're in here. So far the available testing can tell us if there's an active infection. So if it's a positive, it's almost certainly a positive. But they cannot tell us whether we're contagious to others. They're not very useful if you get tested right after you have been exposed within a day or two, or 5 days after the onset of symptoms. They can't tell you if you're going to become symptomatic or remain asymptomatic, and they cannot tell you whether it's safe to assume that you're negative and visit, go and stay with other people and feel certain that you're not going to infect anybody else, particularly if you're not wearing a mask or doing the typical precautions. Is that accurate?
Oh, absolutely. Yes.
Okay. So let's just, let's play this out a little bit. So is there any way that you can see just as a thought experiment, let's say, and this is quite unrealistic, but I just want to do it as a thought experiment. Let's say somebody had access to, PCR testing and they could get results within a few hours. Is there a way that testing could be used to generate, a fairly high level of safety or certainty that you're not infected? Let's say you get tested a couple days before you leave, you drive to your destination, you get tested again when you arrive, which, 2 days after the first test, and both of those tests are negative. What would this sort of level of confidence be in your mind if both of those tests were negative?
I mean, part of it depends on, do you think you've been exposed?
Right.
So there's sort of a kind of a Bayesian statistic element here. This notion of priors. If you've had contact with someone who is now sick, if you've done something, that is high risk, like, go hang out at the bar all night, then, you've got prior information that would indicate that you're higher risk, and therefore you should have less confidence in those negative test results. But I'll tell you something else that I have seen and heard multiple times now, which is this idea of test shopping. There was a Facebook thread and a private Facebook group that I followed back over the summer, where it was these two wedding planners. So they run a professional wedding planner business.
Mm-hmm.
And so one of the bridesmaids tested positive. And so they went on the forum asking if people knew of other places they could get tested, because they need to to show a negative test so that they're allowed to go to the wedding. And so they already have a positive test, and they want to keep taking tests until they get a negative, and then that's what they're going to show to be able to justify being in the wedding. And I've heard this a lot, this idea of test shopping. And the deal is, as soon as you get one positive, you're positive. I'll throw one other thing in here too, that is one of the endless conspiracy theories that seem to go around about some of this notion that you don't really have the virus, you just have leftover components. you have viral
Right.
debris.
You're positive, but you're not contagious.
Well,
That's some of what
well,
people say.
there's a positive but not contagious, but then there's also a positive. This is the whole case-demic nonsense that
Right. Right.
all of those positives aren't really measuring actual live virus. Well,
Just fragments.
yeah, just fragments. The deal
Mhm.
is, I mean, one, it's hard to know for sure. But one of the things that is well understood about RNA viruses is that living organisms do not like to see RNA outside of a cell. RNA has one place it's supposed to be. It's inside a cell, and it shouldn't be hanging around for more than 10 minutes. It exists for very brief periods of time as the DNA is transcribed into the RNA, which is then immediately used is immediately translated into proteins. And so the body is flooded with these enzymes called RNases, whose sole job it is to take any random RNA molecule and, within minutes to an hour or so, slice these things into nuts and bolts. So the idea that you're going to have, stray RNA laying around on top of your throat for, a month, that's unrelated to live virus is really nonsense. That if you've got RNA, the chances are that whatever produced that RNA, was replicating within the last 2 or 3 hours. Now, one of the things that is suspected is that people are able to maintain long-term low-grade latent infection, meaning that there's a small, maybe a small patch that's continuing to replicate virus at a very low level, but not quite at the level that it's going to draw the attention of the immune system. Especially with upper respiratory viruses, the immune system has a hard time effectively reaching the upper respiratory area. And so there's this idea of the virus still being there and still replicating. But if you get a positive on one of these tests, you almost surely have had actively replicating virus in you within the last few hours. So this idea that a test positive doesn't really indicate that you have the virus is nonsense. You've got the virus.
Let's break this down because we're coming up to the end of the time
Right.
here, and I just want to, I wanted to get this out before the holidays. I mean, we missed Thanksgiving, but hopefully, can get this out before Christmas holidays so that people can make more informed decisions. Certainly, different people have different levels of risk tolerance. Different people are at different levels of risk with COVID, due to whether they have their age, whether they have pre-existing conditions, et cetera. And people are going to make choices based on that. But I've heard from quite a few people, friends, colleagues, et cetera, who were planning to use testing in a way that is not really supported by the evidence. So, for example, a colleague of mine was going to they have a school pod, and they were going to try to purchase an Abbott Binax Now test to screen all of the parents and kids every day, every day or every other day or every third day or whatever, even when they're asymptomatic. But as you've made abundantly clear, that's not going to be an effective strategy because you have up to 70% false negative with people who are not symptomatic using the antigen test. So, I wanted people to get a better sense of what the shortcomings are of this testing so that it doesn't give them false confidence. They don't make a plan to go see their family and use one of these rapid tests as a way of ruling out infection and then find that they were positive and that they actually ended up infecting a family member, particularly one who was, at high risk. I really don't want that to happen. And so that's the impetus for putting this out there as soon as we can. What are the testing methodologies that are either in development or maybe close to being released that could help us to address some of these shortcomings and actually provide useful, actionable data?
I think in general, it's dangerous to rely on testing as the cornerstone of a process of trying to remain safe. And there's sort of four words that encapsulate that: White House Rose Garden. There's no population in this nation that has been more intensively tested than the White House staff and the visitors to the White House. That they were running these machines. They were using the Abbott ID Now rapid test. They were running these machines nonstop. Every single person entered the White House got tested. They weren't allowed to see anybody until the test results had come back. Every person in the White House was tested every day when they came in. Nowhere on the planet has there been a more intensive testing process. And what we've seen is because they relied entirely on testing, so no masking, no distancing, no sort of mitigation, other than the testing, you've seen now multiple super spreader events. We're watching one right now with Trump's legal team. Um, and I think that this is probably the single best example, is that whatever, if you've got a school, whatever testing program you're going to dream up is going to be a fraction of what the White House was doing. And it didn't, it has not prevented the White House from seeing multiple super spreader events. So you simply cannot rely on the testing. And I think you're far better off to rely on this idea of prior knowledge about the people and their activities that are coming together. Again, have someone been exposed to a sick person? If they're running a fever and coughing, you may think it's a cold, but you have to assume that it's COVID.
Right.
Have you engaged in activities that are high risk? Do you go to restaurants and bars on a regular basis, or to the gym on a regular basis? Whether you have a mask on or not, you're now in a significantly higher risk category. So a lot of this is, what sort of behavior have you engaged in, and what sort of people you've been exposed to that would give you an indication of your general risk level. But what you see in events, and again, the White House Rose Garden event is a classic example. There was probably only a single super spreader at that event that ended up passing it to quite a number of people. And the problem is that the entire group is only as safe as the least safe member. If you've got 40 people who wear a mask, never go to a store, never go to a restaurant, and never go to a bar, and never leave their house, and you got one person, who hits the bars regularly, that entire group is now, at a risk level equivalent to that one person that was engaged in the riskiest behavior.
Especially if that person happens to be a super spreader, right? And is spreading more virus than someone
Right. And you
else.
may luck out. You may, maybe they're infected and they're not going to infect anybody, but you don't know. You always have to assume that the person you're looking at is potentially infected and potentially a super spreader. So again, I think the assumption has to be that people are infected and you have to take measures. You have to do mitigation. And one of the things that you see there are a lot of clinics, for instance, around the country that have done a really good job. So these are sort of walk-in clinics, emergency care clinics, or like psychiatric service clinics, these sort of things that aren't handling these sorts of cases on a regular basis, but have managed to have very good track records of not having infection within the facility. And I have a close friend who who's a senior executive in a company that runs a series of psych clinics on the East Coast. And every person there takes it seriously. Everyone gets tested once a week. Everyone wears a mask. Everyone follows very specific protocols, because, their assumption is absolutely everyone's infected. If we behave like they are, that's our best shot at no one getting infected. That sort of kind of paranoid mindset is useful. If you think there's a risk, isolate or quarantine. If you've got symptoms, isolate. If you engage in high risk activity, let people know, and know who you're inviting to events. Now, you ask about sort of future technology. So there's a number of really interesting changes that are underway in the testing environment, new technologies that are coming out. So one of the issues that we've seen is just the sheer volume of testing is historically unprecedented. Never, in medical history have we run this many tests for a single infectious disease every day across the country. And so part of what we've seen is a real backlog in the labs in terms of just all of the manual work that needs to be done to set up, prepare the samples, get them on the machines, get the tests run. And so there's some really interesting stuff with what's called next generation sequencing. So these are these genetic sequencing machines that can sequence an entire genome in the matter of a few hours. And so Illumina is the big company that produces these. Their sort of flagship product is called an Illumina NovaSeq, and it allows you to do genetic sequencing on massive amounts of DNA in one shot. So there are companies that are working on using that technology to massively parallel testing of tens of thousands of samples in a run. So the idea is you could put 25, maybe 50,000 samples on a single machine and run this thing, in a matter of 6 hours, and be able to get a result. And each sample is testing, it's essentially molecularly tagged DNA barcoding. We basically attach little tiny molecular sequences that barcode the molecules from each patient sample so that you can then separate everything out at the end in software. So it's a very clever idea, but it potentially is a way to massively increase the volume of testing that can be done.
So would that have roughly, the same sensitivity and specificity of PCR, but just be faster? That be a test more samples at a time?
I mean, it would potentially have much lower false negative rates because you're actually, instead of looking for, say, a 50 nucleotide piece, you could be looking for multiple different segments if you find any one segment, because you're essentially sequencing everything in this process.
Right.
There's also another interesting area of development called metagenomic-based next generation testing. And so the idea here is you take a sample and you basically sequence all the DNA in the sample or all the RNA in the sample. You push the data into software and then you subtract out everything that's human. So you throw away all the human sequences and you look at only what's left over. And then you start looking these sequences up in a master reference database of all known sequences. And the advantage of this is that you could potentially detect a whole panel of different diseases in a single test run. So you could test for every possible strain of cold, of flu, of all the other major upper respiratory viruses and COVID. And so you could come back, with a single sample and a single test, and you could see, someone has a fever and a cough. What is it? This thing would then come back and tell you exactly what it is. If you've got more than one infection, it could see that. It could even see different genotypes, so different genetic mutations of the same virus. So one of the clients I've done a fair amount of work with in Cambridge is developing this technology for the human transplant market to be able to screen transplant patients for various types of viral infections and be able to basically see everything in the sample. But the most interesting stuff is based on CRISPR technology. So this is the gene editing technology that's been in the news a lot. And in fact, the two inventors of this won the Nobel Prize in Medicine back last month. So I'm actually doing work with the company that those two Nobel laureates founded a couple of years ago. And one of the things they're working on are CRISPR-based test platforms. And the deal with CRISPR is that it's this enzyme that does two things. It identifies a genetic sequence, and then it has molecular scissors that will then slice that sequence apart. And so you can adapt those functions to do diagnostic testing. So you engineer the enzymes so that they will detect a specific sequence from, say, the SARS virus, but instead of clipping the genetic sequence from the virus, it instead reaches over and clips an unrelated DNA sequence that releases a fluorescent molecule into a solution. And once it's clipped enough of those fluorescent molecules, and you actually get this sort of lawnmower effect where a single DNA sequence from the virus triggers the enzyme, and then the enzyme undergoes what's called transcleavage, which means it just starts slicing everything it sees within reach. And so it starts slicing these reporter molecules that are surrounding it. Again, sort of like mowing the grass. And so you could then have tens of thousands of fluorescent molecules then released into the solution that can then be detected with the photosensors. So it's a very interesting technology. Again, a completely different way to do amplification. In this case, you're essentially amplifying the number of detector molecules that are present, that are available to be seen from a single detection. The advantage with CRISPR is that it has the potential to be extremely fast, meaning potentially a 5-minute test, very inexpensive, and with both very, very high sensitivity and specificity. So it can be very, very specific, but it can be extraordinarily sensitive. So potentially you could have something with these CRISPR tests that is significantly better than the best PCR test.
But could be done at home and quickly.
And could be done at home and inexpensively. So so in many ways, the CRISPR diagnostics, and this is a very emerging area that is, the earliest work on this is only 2 or 3 years old.
Right.
But the CRISPR diagnostics has the ability to basically break the no free lunch rule, where you can have something that's both cheap, fast, and very sensitive and very specific. So there are products that are developed right now that are very interesting that would be a single-use, disposable, over-the-counter product. There's already been a number of press releases done by companies that are working on this. So Glaxo is very involved. So the GlaxoSmithKline, their home care division, is very involved in this. There are a number of companies that are CRISPR specialists that are working on these sorts of tests. But the goal would be to have a cheap, disposable, 5-minute over-the-counter test as you go down to CVS and buy, and be able to pay, $10 for, $20 for, and have the company still be able to do these profitably. Don't know if we can get there yet. There's both a lot of engineering challenges and a lot of biochemistry challenges that still have to be worked out. But I think it's very promising technology, and it has the ability to potentially supplant all known diagnostic testing technology.
Wow.
So
that's pretty phenomenal. And like you said, like finally possibly breaking the free lunch rule. And that sounds like a test that could make a significant impact on the pandemic, if it actually is possible to realize that in time.
Yeah, and we're probably 6 months out from the first of these going to market. It's hard to know. There's a lot of money, being poured into this. So there's sort of no shortage of resources, talent working on it, but these things take time. But one of the things I think you're going to see is 10 years from now, you probably are going to see very little in the way of PCR still in the clinical environment. And you're probably going to see 2 different types of tests. You're going to see these CRISPR tests that are going to be your sort of everyday, almost everything will go to point of care, where you'll be able to have a small device that you can run, immediately at, wherever the patient is. And then these metagenomic next generation sequencing tests that are able to basically take one sample and find everything within the sample, all pathogens within the sample, which is a very powerful technique. There's nothing quite like that currently on the market.
No, as a clinician, I can tell you that I will be waiting with bated breath for that day to come. Dale, I'm so sorry I have to wrap it up now.
Okay.
I gotta move on, but thank you so much for doing this. It's been incredibly helpful. And where can people find out more about your work? You have an excellent Substack that I follow, and then I understand you've recently launched your own podcast.
Yes. So so I have a newsletter on Substack that's just called COVID Updates. And the way to reach it is just Dale W. Harrison, my name, .substack.com. So DaleWHarrison.substack.com.
We'll put a link to that in the show notes, as well as to the podcast. And where do people find your podcast?
And so the podcast, which I've just launched, and there's only one episode up, but there'll be more shortly, is called The Business of Life Science. And it's a much more specialized podcast that is basically looking at the commercial development side of the biotech industry. So that's a lot of the work I do is things that aren't in the lab
Right.
that are important for these biotech companies to be able to know what products to create and then how to bring those products to market. So it's called just The Business of Life Science, and there
Great.
should be new episodes coming soon.
Yeah, we'll
But
put a link to that as well in the show notes for anybody that might be interested. So thank you, Dale, again. And yeah, we'll have you back in maybe 6 to 9 months' time if the CRISPR technology is getting closer to being realized, and we can talk a little bit more about that. But really appreciate you taking the time to do this. And I think it's going to help a lot of folks who are thinking about the holidays and how to plan for that.
well, thanks. I really appreciate the opportunity to come on and talk. It's obviously a very interesting subject with a lot of detail, and I think there's a lot of misunderstanding as to how these things work certainly how they work in the real world. And and unfortunately, I think a lot of people are making incorrect decisions,
uninformed decisions,
simply because they just don't understand the details and the complexity involved,
the limitations
in this area.
All right. Well, thanks everybody for listening. Keep sending your questions in, chriskresser.com/podcast question, and we'll see you next time. That's the end of this episode of Revolution Health Radio. If you appreciate the show and want to help me create a healthier and happier world, please head over to iTunes and leave us a review. They really do make a difference. If you'd like to ask a question for me to answer on a future episode, you can do that at chriskresser.com/podcast question. You can also leave a suggestion for someone you'd like me to interview there. If you're on social media, you can follow me at twitter.com/chriskresser or facebook.com/chriskresser.L.AC. I post a lot of articles and research that I do throughout the week there that never makes it to the blog or podcast, so it's a great way to stay abreast of the latest developments. Thanks so much for listening. Talk to you next time.